In this video, we're going to be talking about phylum protozoans. In the last video, we introduced ourselves to invertebrate zoology, and we said invertebrate zoology is a branch of zoology that studies invertebrate animals, that is, animals without vertebral column. And we said the most primitive animal that we're talking about first is the animals in phylum protozoans. So today we're going to talk about the biology of the phylum, the representative members of phylum protozoans. Now, starting from where we stopped last week, we have the four classes of phylum protozoan, and that is class Sarcodina, class Mastigophora, class Celiophora, and class Protozoa. And if I could remember, I said representative member of class Sarcodina is Amoeba. Representative member of Mastigophora is 2. We have the Trypanosoma, that is Zoomastigophora, and the Euglena, that is Phytomastigophora. While for ciliata or Celiophora is Paramecium, while for Sporozoa is Plasmodium parasites. These are the members that we'll be dealing with in this video. Moving on, let's talk about the general characteristic of the phylum protozoan. Now, they are mostly unicellular. That is the first thing that you have to note. They are unicellular, they are acellular or non-cellular. They are the most primitive in the animal kingdom. Remember I said their name is derived from two Greek words, that is proto and zoo, that is protozoan, which means first animals. So they are the most primitive in the animal kingdom. They are mostly uninoplates, having only one nucleus. While some can have more than one nucleus, like Paramecium caudatum, which have two nucleus. They are mostly free-living, like amoeba, while few are parasitic, like Trichomonas vaginalis, and amoeba is solitica, and other ones like that. While some are symbiotic and some are commensals, like Tenax and Entamoeba coli. Nutrition may be autotrophic, or autotrophic is also termed holophytic. So they can also be holophytic or heterotrophic, which is holozoic. You understand? So they can either be holophytic or holozoic. That is, they can either feed like plants or they should feed like animal. You understand? Now, going from there, they are mostly aquatic. That is, they are what? They are found in water. They are mostly aquatic. That is, they are found in what? In water. And they can exhibit both sexual and asexual reproduction. We talk about the sexual reproduction in Paramecium, where we talk about conjugation of what? Of Paramecium. Now, the first member that we'll be taking tonight is the class Mastigophora. Let's talk about the general characteristic of the class Mastigophora. They are sometimes called the flagellates, and this is because their organelle of locomotion in adult mastigophore is flagella. They possess flagella, which I'll be showing in this. Look at this. This is their flagella. So they possess the flagella for organelle of locomotion. Now, going back to our slide, their body is covered with pellicle, and this pellicle is what makes them to assume a particular shape. Understand? They have a particular shape, and they are mostly free-living, while few are parasitic. Free-living one is Euglena, and the parasitic one is what? Is Trypanosome. While I said they exhibit holophytic, which is behave like they feed like plants, or holozoic, which means they feed at what? They feed like animal. Now, binary fission is longitudinal. That is, whenever they want to divide, they divide along the horizon. You understand? I'll still say that when we get to the life cycle of mastigophore. They are further classified into two. That is, they are either subclass Phytomastigophora or subclass Zoomastigophora. So the Phytomastigophora are the one that behave like plants, and the zoomastigophora are the one that behave like animals. And the reason why they behave like plants is because there is a presence of something we call chlorophyll. That is why they behave like plants. They have chlorophyll. While this one have no chlorophyll. So I mark absence of what? Absence of chlorophyll. So that is why we said they behave like animals. Now, moving from there to biology of Euglena. Biology of Euglena. Now, the biology of Euglena, we have to understand that Euglena is a plant-like mastigophore, and this is a representative of Phytomastigophora. They are mostly found in water, in ponds, that is in ponds, and in stagnant water. They have their anterior end to be blunt and their posterior end to be pointed. Now look at their structure. We have the anterior end to be blunt and the posterior end to be pointed. That is what we mean by that. They have the outer layer to be ectoplasm and the inner layer endoplasm. And they have two flagella. They have two flagella. That is the short and the long flagella. So inside this space, we have the short and the long flagella. We have the short and the long flagella. So checking here, we have a short flagella inside this space and a long flagella, the one that is protruding outside. That is the structure. And this flagella arises from a structure known as kinetoplast or kinetochore. Arises from kinetoplast or kinetochore. I think it was not indicated here, but we have something known as kinetoplast. Kinetoplast. That is where they indicate from. That is where they originate from. Now, the endoplasm of these animals has what? Has nucleus. It has nucleus. It has Golgi apparatus and it has contractile vacuole. Let me print the ones that I made before so that it won't cause confusion. That's what? It has nucleus, it has contractile vacuole, and it has the Golgi apparatus. Now, the contractile vacuole is used for osmoregulation. That is what they use contractile vacuole for. It's used for osmoregulation. And there is something we know as a stigma or eye spots. Remember that I said they are autotropic. And when something is autotropic, it means it can undergo photosynthesis. That is the reason why they said it is autotropic. Are you getting it? Now, since it is autotropic, it needs something to sense to let it know where there is light. You understand? So this sigma or red eye spots, the eye spots, it is used for sensing light. It is a photoreceptor. So I call it a photoreceptor. I'll call it a photoreceptor. So it is used for sensing light so that they can know where they can move to, so that they can produce food by themselves. They are what? They are holophytic. So this is the description of what? The um description of Euglena. Now, what about their excretion and respiration? They excrete through simple derivation, just a simple derivation. That is how they excrete. So they have no organelle for excretion. They have no special organelle for excretion. So it's through the what? It's through the uh, body surface. I get me now. So that is where they excrete, and it is by what? It is by simple diffusion. Now, talking about the nutrition, they exhibit autotrophic mode of nutrition, like I said. Hence, they are what? Holophytic and produce their own food by themselves using photosynthesis process in the presence of what? In the presence of sunlight. And this is because they have chlorophyll. They have what? They have chloroplast. That is, the chloroplast secretes chlorophyll, right? The chloroplast secretes chlorophyll. And the chlorophyll is used for what? Uh, it's used for photosynthesis. Hence, they got their name Phytomastigophora due to this particular characteristics. You understand? So because of this characteristic, that is where they got their what? The name Phytomastigophora. Are you getting that? So because they behave like plants. Now, let us now go to their reproduction. Now check this out. This is how they reproduce. This is what we know as longitudinal fission. Now if you notice very well, it divides along the axis, like it divides vertically, not horizontally. this is the first step of division. Take have another image here that describes that. Yeah, this is another image. Can you see? Can you see the way they divide? Now look at it. This is the first stage. This is the resting stage. Now, if you look at the second stage, you will notice that uh, the nucleus has become two. Let me change the color. That the nucleus has become what? The nucleus has become two. Can you see? Let me put that so that you can see what the nucleus has become two. So now, as the nucleus divides in this third stage, the cytoplasm, look at this line. The cytoplasm begins to what? To divide vertically. You get me now? To divide vertically. Now, when the cytoplasm started dividing vertically, we notice that what? The this one is just a line, just showing that it starts dividing. Where in this case, the cytoplasm divides, they separate from each other. One goes like this, the other one goes like this. You understand? And we are now having two daughter euglena. I get me now. So this is what? This is just a longitudinal vision of euglena. Longitudinal vision of euglena. Now, that is what we have for euglena. But this is asexual reproduction. Asexual, with no record of sexual reproduction. They have no record of sexual reproduction. That should be noted. Understand? Now, moving from there, this is another illustration of their reproduction. This is a longitudinal feature. So I will go back to Lena. Uh, so this is just a note that you can actually jot down from there. We have all this, you see. So that is that and that. That is everything I've explained just now. Now, moving from there to the next one, that is biology of Trypanosoma brucae. And this living organism is a representative of what? Representative of Zoomastodophora. Trypanosoma are unicellular, parasitic, flagellated protozoans, and they belong to the family Kinetoplastiae. Now, the word trypanosoma has been taken from two Greek words, that is trypano, bora, and soma, which is body. So they bore into the body. Are you getting me now? Now, they are exclusive at flagellates, like I said, because they possess of what? They possess flagella. Hmm? Now, they lack chlorophyll and they are obligatory intercellular parasites. Now, I can describe them as being heterogeneous. As being what? As being heterogeneous. Because I describe them as being heterogeneous because they exist in more than one host. That is, they have two hosts. And the host is Tsetsefly, and human or cow. That is, there are two hosts. So they have two hosts. They have the primary host and the secondary host. Now move from there. We now have the morphology and the life cycle of what? Of this group of animals. The morphology and the life cycle of them. So this is the morphology and life cycle, which I will be describing. Now, one of the things that they cause what we know as African sleeping sickness. So they cause sleeping sickness. That is what they cause, sleeping sickness. Now, look at their structure. This is their structure. They have a flagellum. You understand? They have a flagellum for their movements. Are you getting that? They have an ectoplasm and they have the endoplasm. The endoplasm consists of every of their organelles. The endoplasm consists of it, just like a cytoplasm. They have the nucleus, which is the life of their cell. They have a nucleus, which is the life of a cell. They have a nucleus, which is inside the nucleus. The undulating membrane, which surrounds the what? The organisms. And on the undulating membrane, the undulating membrane is formed by what? By a flagella. And the flagella is attached to the body of the animal. I get me now. I now have the blepharoplast or the basal ganglia. Now look at this. Let me see. Let me clean all this so that I can actually uh, get that right. I need to explain something in this blepharoplast. Now look at this. Look at this blepharoplast or basal ganglia and kinetocha. I see that. Now these are the origin of the flagella. If you look at it very well, at it. These are the what? These are the origin of the flagella, the blepharoplast and the kinetoplast. I get me. So the two flagella originate from the what? The blepharoplast and the kinetoplast. So that is their morphology. Hmm? Now, their what about now? Another thing that you have to notice here is that they have no wood vacuole. They have what? They have no wood vacuole. The the cytoplasm only consists of nucleus, parabasal body, and the kinetoplast and the kinetochore. I get me now. They have no wood vacuole. That's another thing that you have to note. Now, they exist in two hosts, like I said. They exist in what? In two hosts, and this is their what? This is their life cycle. Sorry, I should answer that. And this is their life cycle. Look at their life cycle. The tsetse like bites man. Can you see? Tsetse bites man, and the infected metacystic trypanosoma figure transfer into the bloodstream, which is carried to other sites. It's carried to other sites and multiply by what? Multiply by binary fission, which means there is no sexual reproduction. They multiply by binary fission. Can you see the way they multiply? Can you see? They also divide, just like Euglena. They divide along the axis, just like Euglena. Now look at this one as well. Divides, just like Euglena, and they separate. Now after separating, the Glossina palpalis, that is the tsetse fly, comes to another man, take a blood meal from the man. Once it takes a blood meal from the man, takes the adult trypanosoma zygote, it takes it in, and once it takes it in, they undergo sexual they undergo another procyclic this thing. Once they enters the flies, when they enter the tsetse fly, they divide into two again. I get me now. Into two again. And once they divide into two, they keep multiplying the salivary gland of the what? Of the fly and transform into a metacyclic trypanosoma zygote, which is then transferred back into another human. And this is how the life cycle proceeds. This is how the life cycle continues. So that is that on trypanosome. That's that on trypanosome. Now look at, we are now moving to biology of amoeba. Amoeba creatures. Like we all know from secondary school, we know that amoeba is something that has no shape, right? It has no shape. And why does it have no shape is because it has no pedicle. The reason why they do have any shape is because they have no pedicle. Amoeba are mostly found at the bottom of the freshwater. So this is just a little jetting from amoeba. Let me show you the structure. So this is the structure of amoeba. They are mostly found at the bottom of the freshwater. This is the microscopic structure of amoeba. Yeah, this is the microscopic structure of an amoeba. They are found in what? In the freshwater at the bottom. And they have a lobe shape. And they have a lobe shaped structure called pseudopod. That is their pseudopod. That is what they use for movement. Their pseudopod. That is what they use for movement. And they have a thin structure called the cell membrane. They have a what? A thin structure called the cell membrane. Can you see it here? They have the cell membrane here. Plasm is divided into two. Into what? Into two. That is the ectoplasm and the endoplasm. So they have two like the cytoplasm. It's made up of two parts. That is the ectoplasm and the what? The endoplasm. And the endoplasm contains all the what? Contains all the cellular organelles. Understand? Including the food vacuole, the nucleus, the crystals, and other things like contractile vacuole. You understand? Now, they feed by an action called phagocytosis. Phagocytosis. Phagocytosis, which means they engulf food materials. They feed on algae, they feed on diatoms. They engulf food material using their pseudopod. You understand? So this pseudopod is used for what? For food movement and what? And feeding. That's why they call it pseudo. Pseudo means false and pod means leg. So that is false leg. Now this pseudopod is two. We have the lobopoda, lobopoda and the philopoda. The lobopoda and the what? And the philopoda. These are the two types of what? Two types of pseudopod that we have. I'm going to rewrite that again soon. Now, continue. Any food that they engulf using their what? Using their pseudopod. Any food that they engulf goes into the cytoplasm and it is stored inside the what? It is stored inside the food vacuole. You get me now? So it is stored inside the food vacuole. Now, the waste material is passed out of the body by simple division, which means they also excrete by what? By simple division. Contractile vacuole is used for osmoregulation. They use contractile vacuole for osmoregulation. That is for what? For taking out water of the body because they are living inside water. If they take in too much water, they have to excrete it out. So they use contractile vacuole for what? For osmoregulation. Like I said, I'm going to write something that is these two things: lobo, lobopoda, lobopoda, and philopoda, philopoda. So these are the two aspects of pseudopoda. We are having two types of pseudopoda: the lobopoda and the what? And the philopoda. Now, moving from there, they have no anus. I get it now. They have no, they have no anus. So the pseudopod also serve as what? As anus, in which they use for what? For excreting food materials. Remember, I told you that they use the body surface, and the pseudopoda is always on the what? It's on the body surface. So they use the body surface for what? For excretion as well. So these animals they reproduce in two ways. They reproduce by binary fission and they reproduce by multiple fission. By what? Binary fission and multiple fission. Now this is the structure of them. Now look at their reproduction by binary fission. Very similar to um very similar to that of the glanna. But if you notice very well, their own division occurs at the what? Occurs at the along the axis. Can you see how they divide? Can you see? The nucleus stretches, and once the nucleus stretches, it breaks into two. We have one cell here, we have the other cell here. Then at the end of the day, they do what? They separate into two daughter amoeba. That is binary fission. I get me now. Now look at the multiple fission. This multiple fission occurs in um in unfavorable condition. Unfavorable condition. That is where the multiple fission occurs. Unfavorable condition. For example, maybe the water was poisoned, or the water becomes dry, or maybe something happens to the water. Maybe a chemical is dropped in the water. So that's an unfavorable condition. That is when they undergo what? A multiple fission. Now, how do they undergo multiple fission? They keep dividing inside themselves. They keep dividing the nucleus without separating the what? Without separating the cytoplasm. That is multiplication. Now they now form what we know as energid nucleus. Energid, energid nucleus. That is what they form. And the energid nucleus is also known as cyst. Understand? It's also known as cyst. So they form what? Energid nucleus. Spelled ENERGID. Energid nucleus. That is what we have there. Now, once the condition is now favorable back, like once the condition is favorable back, then it bursts. The cytoplasm bursts to release small amoeba. Can you see? Can you see the one that I'm marking? Release what? Small amoeba. So this nucleus is known as what? It's known as energid. Let me write that again. Energid nucleus. And this only occur under what? Under unfavorable condition. This binary fission occurs normally, while this one occurs only under unfavorable condition. Now, moving from there to biology of paramecium. Paramecium caudatum. They are present in the phylum, this class Celiophora, because they are mostly found in brackish water though. They are mostly found in brackish water. That is, they are aquatic. Their body is covered with pellicule, hence they have a specific shape. They are also called slipper animalculae because they look like slippers, and their body is distinctly differentiated, which makes them the most complex protozoan. The most complex protozoan is what? Is the paramecium caudatum. I get it now. So other protozoa are not as complex as paramecium. The protoplasm is divided into ectoplasm and endoplasm, and they have something we know as trichocyst. The trichocyst is used for defense and anchorage. I get me now. They use trichocyst for defense. They have a pellicle which is elastic, and the pellicle gives them the shape which is a slipper shape. They have food granule, that is they reserve food inside the what? Inside the food granule. And some of the granule are secretory or excretory, which means they have the reason why I said they are the most complex is because they have something we know as cytoproct, and the cytoproct acts as anus, like an anus, you understand? Now they have two nucleus, that is the micronucleus and the macronucleus. That is the two nucleus that they have, the micronucleus and the macronucleus. I get me now. Now, the macronucleus is used for during reproduction. You understand? So I'm still going to talk about that used during reproduction and what's micronucleus used for as well. So like I said they have what? They have a mouth, and the mouth is known as cytosome, and they have an anus, and the anus is known as cytoproct. They have numerous food vacuole. I'm going to show you the structure now. So this is the structure. They have numerous what? Food vacuole. This is their contractile vacuole. The contractile vacuole is used for osmoregulation. I get me now. This is the micronucleus, used for what? Used for reproduction. This is the macronucleus. This is the cytoplasm. We know what the cytoplasm is. A jelly-like body that contains all the what? That contains all the organelles. This is the food vacuole where they store their food. This is the pellicle, what gives them shape. This is their cilia that they use for what? For movement. This is the oral groove where food enters. This is the buccal overture where food passes. And this is the cell mouth, that is where they use for eating. And this is the anal pore. The anal pore is for what? For excretion. So this one is used for excreting water. This one is used for excreting what? The waste product that is not needed by the body. You understand? So that is what? That is for structure of paramecium. Can you see that they look like a slipper? So they are called slipper animalculae. Now, like I said, they have a trichocyst. Trichocyst is not indicated here. They have a trichocyst. The trichocyst is used for what? Is used for anchorage and defense. And the oral group is for what? The oral group is for feeding. I get it now. Now, the organ of locomotion is cilia. The organ of locomotion is cilia, and they are found in what? They are mostly found in brackish water. Now, they are the most complex protozoan. And how do they reproduce? They reproduce in two ways. In two ways. The two ways is, remember I said this one is cytoproct. Cytoproct is anus. Cytosome is mouth. Please don't forget that. Now, they have the what? They have a mouth and the anus. Now, just talk about the reproduction in what? I've talked about the nutrition. Let's now talk about the reproduction in this. They can divide two types in two ways. They can divide uh by binary fission, that is asexually. They can divide sexually. During asexual reproduction, the cell divides, you understand, and the mic the micronucleus divides mitotically, while the macronucleus divide amitotically. Please, that should be noted. The micronucleus divide mitotically and the macronucleus divide amitotically. I'm going to show you the structure. Now, look at the structure here. We have the structure. Look at it. We have sorry, I have to change this. Now look at this. This is the micronucleus. This is the what? This is the macronucleus. Now look at the micronucleus dividing mitotically via mitosis. Can you see the line? Something like a mitosis. Why the macronucleus just dividing, like cutting into two? Which means during division, the micronucleus is going to transfer gene, like going to share chromosome. You understand? That is mitotic system. Now, during reproduction, oral group disappears. You see, the oral group disappears. While once they started dividing, the oral group started appearing again in two what? In two ways. Then can you see that the cytoplasm starts cutting along the line? Can you see it starts cutting? And once they cut, they divide into what? Two paramecia, two daughter paramecia. See, two daughter paramecia. So that is what? That is their division. While their sexual reproduction is by conjugation. Sexual reproduction is by conjugation, and in this process, the conjugation bridge is formed when two paramecia unite. And these two paramecia that unite are known as conjugates. Please note that. They are known as what? They are known as conjugates. And they have, let me show you the structure. Look at this. Look at this. So this is two paramecia coming together. What is it? This is two paramecia coming together. Hmm? Now, at the end of the day, we start exchanging materials. We start exchanging genetic material. Now, after exchanging genetic material, they undergo what we know as syngamy. You understand? During syngamy, they now have what? We now have, look at it, count 1, 2, 3, 4, 5, 6, 7, 8. They are going to have 8 nucleus. That is diploid nucleus. And look at it, these ones are going to fuse together to form a single micronuclei. And these ones are going to come together to form a single macronucleus. And this is going to divide into what? Into two daughter paramecia. That is what? That is conjugation of what? Of paramecia. You understand? So that is that on paramecia. Now, going from there to sporozoa. Sporozoa, we have the Plasmodium species. Plasmodium. I did not really write note about that. So this is the structure of Plasmodium. This is Plasmodium parasites. Now, we have four types of Plasmodium. And Plasmodium causes what? It causes malaria. We have four types of Plasmodium. The first one is Plasmodium vivax that causes partial malaria. We have Plasmodium malariae that causes quantia malaria. And we have Plasmodium falciparum that causes pernicious malaria. Understand? Now, the life cycle of Plasmodium is quite different from the life cycle of others because they also have two hosts. That is the main host and the definitive host. That is the primary host and the secondary host. And the primary host is known as mosquito, and the secondary host is what? Is man. Look at it. Now, this is how they start their life cycle. A man is they have three cycles. They have three life cycles. They have the exoerythrocytic life cycle, or known as pre-erythrocytic life cycle. They have the erythrocytic cycle, and they have the what? And they have the sporogonic cycle. These are the three this thing that they have. Now, talking about exoerythrocytic life cycle. Now, a man is bitten by a mosquito. A man is what? Bitten by a mosquito. It is inside the bloodstream that it's going to release what we know as what? What we know as megazoites. It is going to be released into the what? Into the body of a man. Now, once it is released into the body of a man, it looks for a way to move into the what? Into the liver. I get me now. They move into the what? They move into the liver cells. Once they move into the liver cells, the merozoite develops into what we know as schizons. And this schizon is the main problem of malaria because that is once someone is having schizon like this, it's going to be developing what? The symptoms of what? Of malaria. Now, the schizons develops into immature trophozoites. Then from immature trophozoites to what? To matured trophozoites. I get me that. All inside the liver. Hmm? Now, we now have two destiny. After developing into immature trophozoite, it's either it develops back into schizons, rupture the schizons and release it into the bloodstream, and it starts again. I get me now. Or the mature trophozoite develops into gametocytes. I get me. So into gametocytes. Now, once they develop into gametocytes, they are going to develop into two types of gametocytes. That is the microgametocyte and the macrogametocyte. The microgametocyte is what? Is the male gamete, and the female gametocyte is the what? The female. Now, the microgametocytes, once these two gametocytes is going to be taken by the mosquito. Look at it. Going to be taken by the mosquito during blood meal. They take in blood meal and they are infected by the what? By the gametocyte. Now, once they enters the what? Once they enters the gut of the mosquito, the microgametocyte exflagellates and develop into what? Something like a flagellated microgametocytes, and the macrogametocyte remains the same, develop into microgametes. Now, the both of them now what? The male gametocyte swim to the female gametocyte and they fertilize each other. Once they fertilize each other, they develop into ookinete. Then from the ookinete to oocyst. To what? To oocyst. Now, the oocyst now migrate from the stomach of the mosquito to the gut of the mosquito. Once they go to the gut of the mosquito, they release sporozoites. You understand? Once they release sporozoites into the body of the mosquito, the sporozoites stays there in the salivary gland of the mosquito. Once the mosquito bites a man again, the man is infected what? With sporozoites. That is the life cycle of what? Of plasmodium parasites. Now, I hope you understand this particular aspect. Now, the quick question. Um, Protozoans are mainly unicellular or multicellular. Remember, they are what? They are unicellular organisms, right? And in protozoan, phylum protozoan, um we have how many class? We have four classes. That is class Sarcopina, Mastegophora, Celiophora, and what? And Sporozoa. And this Mastegophora is represented by two. That is Phytomastegophora and Zoomastegophora. The Phytomastegophora is Intena, and the Zoomastegophora is what? Is Trypanosome. The Sarcodina is amoeba creatures. The Sporozoa is plasmodium parasites, and the um, the Celiophora is paramecium. I hope you learn one or two things from this video. Stay tuned for the next video. Thank you. Good night.